All right. So your eye should look like this. And what you're looking at here is a cow eye. So in the orbit, there's quite a bit of fat that is surrounding the eye, kind of cushioning it. And that surrounds these extrinsic eye muscles. So that's the dark tissue that you can see right here. The extrinsic eye muscles, you can kind of tell which way is up and down, medial and lateral on these if you really dig around. I would not ask you to do that, but you could, in theory, figure out which one's which if you spent some time on them. But all you need to know for this is just that these are the extrinsic eye muscles. And then if you look in the back of the eye, you'll see a space. Some of you might have to dig a little more than others, but you have this little kind of hard nub sticking out. That's the optic nerve that's exiting from the back of the eye. So if I go back to the front of the eye, usually it depends on the eye itself, but they get a bit of the conjunctiva. So like this tissue that's kind of, I can kind of see it moving a little bit as I move my probe across it on the white of the eye or the sclera. This is the conjunctiva, that little, usually get a little kind of lifted up a bit with this. So I'm kind of underneath it and lifting up the conjunctiva right now. Some of you might have some of the palpebra on here. So like if you can go down and you can get a, kind of see a pocket right there. You have the layer of the conjunctiva that's on the eye. That's called the ocular conjunctiva or bulbar conjunctiva. And then the layer that wraps around and then comes back up on the inside of the eyelid would be right here. That would be called the palpebral conjunctiva. So it's kind of like a serosal membrane that has a parietal and a visceral layer. The visceral is basically what our ocular conjunctiva is. The parietal would be the inside of the eyelid. So if you bend your eyelid down and you touch that pink part, you're touching the palpebral conjunctiva. And that creates a seal so that like bacteria and stuff and dirt and debris can't get through here and then back into the back of the orbit, which then has a pretty straight shot to the brain, right? So when we're looking at this, you'll notice the conjunctiva does not extend over the cornea because it would disrupt our vision. So the cornea is the clear part. It's collagen, just like the sclera, but it's organized a lot more organized, I guess, arranged in a much more organized fashion, meaning it doesn't disrupt light. And so this is clear. Now, when we preserve it, it denatures the protein. And so it gets a bit cloudy because now the protein isn't as neatly packed. It's all tangled up and loose from the denaturing process. So normally you wouldn't be able to see through this, the same way your cornea looks crystal clear. We'll see the same with the lens too, that it has that same yellowish coloring, and that's because of the preservation, not because of the animal. So you can see through this too. So again, here's the cornea, this clear thing. I can see the pupil right down the middle, and then the dark that rims it is the iris. Now most animals don't want you to know where they're looking. Because if like a predator sees that I'm looking at it, it knows that it was detected and it's going to try and go for me right away. So most animals, their cornea and their iris covers much of the large portion, and they have a pigmented sclera sometimes too. In humans, our vision is a big part of our communication. And so I know that you're listening to me, right? If you look at me, or if I look at you, you know that I'm paying attention to you because you can see the whites around your eye to highlight your iris. We've actually evolved dogs a little bit because dogs, if you look at their eyes compared to like wolves, you can see a lot more of their whites because we like animals that we can tell if they're paying attention to us. So it's kind of cool that we've shifted their evolution in that process. But that's why we won't see, like in a cow, you won't see the whites of their eyes unless they're really rolling their eyes, or horses too, same idea. So where you want to cut on this is find this line right between the cornea and the sclera. Now, I'm touching the conjunctiva, but the sclera is the connective tissue right underneath it. And I'm going to go down, so like on this probe, the bend on the probe is about 1/4 of an inch long. So see how the tip of this is straight and then it bends to the main shaft that I'm holding? So I want to go about that distance, so right where the bend on the probe is, which would be right here now. That's how far down I want to go. And then I'm going to cut in a circle around this. The reason for that is I want to get below the ciliary body. That way I'm cutting into the posterior chamber. So when I lift this off, then I'm able to see the anterior chamber on one side, the posterior on the other. Now it's tough material, And so the way to do this is if you have one of these scissors that have a blunt and a pointy end, you just hold it so that the pointy end is there, and then you push it in, and that gives you a starting place. If you don't have these scissors, or like your scissors aren't that pointy, don't force it. I just grab a scalpel and make a small little poke. Don't use the scalpel to cut around, it's harder. So then if I get my scissors, I can then put that pointy end in there, and then I'm going to just cut in a line staying about that distance. Kind of overshot there. Connect those lines. So now I'm not taking this off yet. So you want to be a little careful when you take it off, because you want to notice a few things when you do that are going to get disrupted pretty quick once you open it up. So I like to just kind of take the probe and I'll just run it through underneath. And what I'm doing is breaking that jello of the vitreous humor apart so it doesn't suck out all at once. So now that I've kind of separated those a bit, I can then lift this off. Set it over there. So you'll see that there's this little jiggly goo. This is your vitreous humor that's in here. And the yellowish kind of opaque, like not quite see-through, but a little blurry thing, that yellowish layer, I can sort of separate just that. That's the retina right there that I'm holding. Now notice that it's not attached to the back of the eye. This is why, like, if you bungee jump or have a violent head trauma, you can detach the retina. Now it does get blood from the retinal blood vessels, but a lot of the supply for the photoreceptors comes from the choroid right underneath it. And so if you detach the retina, those photoreceptors can start to die, which is why they'll try and reattach it. They use like lasers in surgery. The other thing I can notice too, this one has some inclusions in it, is notice how like this all kind of puckers right in the center. I'll try and zoom this video in for the one I post. But like there's like a little point here where all that retina goes in. That's the optic disc. So you're seeing where all those axons exit, and if I were to go and flip the eye over, I'd find the optic nerve right behind that point. So then you can just pour out, or kind of slough out this. Notice as I tip it over, the retina is going to get drugged with the vitreous humor. So that's why I wanted to pull the eye cap off, or the anterior portion off carefully, so I could see how the retina is held against there. Because once I do this, the retina is just going to look like a little booger that's kind of hung on the wall. So I'm just going to kind of coax that vitreous humor out. And now I can see that the retina is just this single layer. And it stays attached back here at the optic disc, because that's where all those axons are exiting out. So I can just kind of move it around and see that. Sometimes if you're lucky, you'll see the fovea centralis as a little, it looks like somebody put a little pin on part of the retina. Sometimes you don't see it. So that's about it for the back of the eye. I can see the tapetum fibrosum here, which is part of the choroid, that pretty blue kind of mother of pearl color. So that's what's going to reflect the light. And that shows you where the tapetum lucidum's at. That shows you where the bottom of the field of the view is for the cows too. The anterior part of the eye has a little bit more anatomy. So let me clear off. I'm going to gently clear off this vitreous humor. Thumb's a little easier for that. It's like a jellyfish that you see washed up on the beach. So notice that the lens, this yellow portion, it's still kind of held in place. And then if I go over here, I can see on this that I have my cornea. The pupil is the circle, and then the iris is the dark. So on the back, what I'm seeing is the iris is this rim that's kind of sucked up right against the lens. Get this more of this fluid out. And I can see a little bit of the retina where I made my cuts along here too. Most of the back of this, and you'll see this more easily in a few minutes, but most of the back of this is what we call the ora serrata and the choroid body. And that's just going to be kind of this like mass that the aqueous humor is produced from. So I'm still in the posterior cavity here. The lens is really the divider between the anterior and posterior cavities. So when you take the lens off, I want you to just take this probe and go under one side of it and then carefully pry it up. And if you watch close, it's probably not going to show up in the video, but if you watch closely, the lens is held by this little tiny membrane that I just tore. Those are called suspensory ligaments, and those are what anchor the ciliary body. I think I called it a choroid body last, but they anchor the ciliary body to the lens. So when my ciliary body gets bigger, it stretches the lens out, and when it relaxes, the lens gets fatter and gets relaxed in the middle. That helps you bend light when you look at stuff. So I can pull the lens all the way out, and I see that membrane just kind of tearing as I do. And you'll notice the lens is kind of cloudy and it's hard. If you were to feel this in real life, it would be super flexible and very clear. But the preservatives make it kind of like a hard little blob. Wish we had some real like fresh eyes so we could see this. But like if you ever played with a contact, you know how flexible and soft they are. So it's similar in that kind of texture. Now once you have the lens out, I think it's easier to cut this front in half. And we'll be able to see a few more things. So now that I've cut this in half, I can clearly see the cornea here is separate from, not separate, but it's a different look to it than the sclera that's very white because this allows light through. And then I see this little ridge running through here. That ridge is the iris, and the hole that like my probe is going through right in this space, that would be the pupil. So I like to ask about that, like identify the space the pen passes through as like that would be the pupil. This space between the iris and the cornea is the anterior chamber of the anterior cavity. And then the little tiny bit that's behind it before you get to the posterior cavity is the posterior chamber of the anterior cavity. And when I clear off this vitreous humor, it's probably not going to show up in the video great because it's just tough to see. But there's actually a little stepped ridge. So you have the iris and then a little bump. And that bump is the ciliary body that was holding onto the lens. And it'll have little frills like the underside of a mushroom on it. And so it looks pretty distinctive. So hopefully that helps. If you have any questions or want to help with identifying stuff, just let me know and I'll come by.